Hello friends, today in continuation to the previous session on cytoskeleton, we will discuss intermediate filaments. Intermediate filaments are found in nearly all eukaryotic cells from multicellular organisms, but their presence in fungi and other lower unicellular eukaryotes is controversial. In epidermal cells and the axons of neuron, intermediate filaments are at least 10 times more abundant than microfilaments or microtubules. In immunofluorescence micrographs, we can see a network of intermediate filaments filling the entire cytosol of a cell in a pattern usually distinct from that of microfilaments but similar to that of microtubules. Here we look at intermediate filaments. These are exclusively structural proteins that are very static. The organization of intermediate filaments and their association with plasma membranes suggest that their principal function is structural, to reinforce cells and to organize cells into tissues. Indeed, the supportive role of intermediate filaments is carried to an extreme in claws and hairs, the dead remnants of epidermal cells, which are composed largely of intermediate filament proteins. Perhaps the most important function of intermediate filaments is to provide mechanical support for the plasma membrane where it comes into contact with other cells or with the extracellular matrix. Unlike microfilaments and microtubules, intermediate filaments do not participate in cell motility. There are no known examples of intermediate filament dependent cells movement or of motor proteins that move along intermediate filaments. Several physical and biochemical properties distinguish intermediate filaments from microfilaments and microtubules. To begin with, intermediate filaments are extremely stable. Even after extraction with solutions containing detergents and high concentration of salts, most intermediate filaments in a cell remain intact, whereas microfilaments and microtubules depolymerize into their subunits. Intermediate filaments differ in size from the other two cytoskeletal fibers. Indeed, their name derives from their 10 nanometer diameter, smaller than microtubules which are 24 nanometer but larger than microfilaments which are 7 nanometer. Moreover, in contrast to the beaded filaments and the hollow tubules formed by the globular, actin and tubulin subunits respectively, intermediate filament subunits are alpha helical rods that assemble into rope-like filaments. Finally, intermediate filament subunits do not bind nucleotides, and their assembly into intermediate filaments does not require nucleotide GTP or ATP hydrolysis, as does polymerization of G-actin and alpha or beta tubulin. However, many of the details concerning the assembly of intermediate filaments in cells remain speculative. Structure of intermediate filaments. In higher vertebrates as we and animals, the subunits composing intermediate filaments constitute a superfamily of highly alpha helical proteins that is divided into 6 major classes or types on the basis of similarities. They are in general grouped as type 1, referred to as acidic keratins, and type 2, referred to as basic keratins of the epithelia as heteropolymeric keratin filaments. Cytokeratins are more generally found in epithelia that line internal body cavity. Type 3 has vimentin present in mesenchyme such as fibroblast and typically expressed in leukocytes, blood vessels, endothelial cells. Desmin are found in muscle, glial fibrillary acidic protein in glial cells and astrocytes. Very little is studied about peripherin protein of the peripheral and central neurons. Type 4 has NFL, NFM, NFH, that is the low medium and higher molecular weight neurofilaments found in mature neurons. The internexine is developing central nervous system. However, there is also a non-standard type 4 called filancin in Lance's fiber cells and facinin, whose tissue distribution is yet a mystery. The last more type 5 has A, B and C lamine present in nucleus of all cells. Unlike the actin and tubulin isoforms, intermediate filament protein classes are widely divergent in sequence and vary greatly in molecular weight. We introduce the 5 classes here and later consider their functions in various cells in more detail. The 6th class has little similarity to the other intermediate filament proteins and is not discussed. Members of this class are found in the lenses of the eye and form beaded filaments. The expression of each intermediate filament protein is characteristic of a certain tissue or a cell type. Intermediate filaments shows species-dependent variations in molecular weight. Almost more than 15 isoforms of both acidic and basic keratins are known. Let us now observe the structure in an electron micrographic view. In electron micrographs, an intermediate filament protein dimer appears as a rod-like molecule with globular domains at the ends. Labeling experiments with antibodies to the N and C terminal domains indicate that the polypeptide chains are parallel in a dimer, a pair of dimers associated laterally into the tetramer. Here is the electron micrograph of a neurofilament purified from spinal cord. After the filament is rotary shadowed with metals, long whiskers are seen in project from its walls. These projections correspond to the N and C terminal domains of the neurofilament protein tetramers. Formation of intermediate filaments. Besides sharing an ability to form filaments of 10 nanometer in diameter, all intermediate filament subunit proteins have a common domain structure, a central alpha helical core flanked by globular N and C terminal domains. The core helical domain, which is conserved among all intermediate filament proteins, consists of 4 long alpha helicals separated by 3 non-helical regions. The positions of these spacer elements are also highly conserved among all intermediate filament proteins. The alpha helical segments pair to form a coiled coil dimer. There are specific level of organization and assembly of intermediate filaments. Intermediate filament proteins form parallel homo or heterodimers with a highly conserved coiled coil core domain and non-helical tails and heads which are variable in lengths and sequence. The central core domain contains 3 non-helical spacer elements. A tetramer is formed by antiparallel staggered side-by-side aggregation of 2 identical dimers. Tetramers aggregate end to end, forming a protofilament. Pairs of protofilament then laterally associate into a protofibril. Lateral association of 4 protofibrils form a cylindrical filament are 10 nanometer thick. Dimers in a tetramer have an antiparallel orientation. Some evidence discussed later suggests that the tetramer and not the dimer is the subunit for assembly of intermediate filaments. Tetramers bind end to end, forming protofilaments 2 to 3 nanometer thick, which pair together into protofibrils. Finally, 4 protofibrils form a single intermediate filament that is 10 nanometer in diameter. The tails extending from the tetramers composing a protofilament often are visible as projections along the length of a fully assembled intermediate filament. Interestingly, because the tetramer is symmetric, an intermediate filament may not have a polarity like an actin filament or a microtubule. This idea is supported by experiments showing that vimentin subunits can incorporate along the length as well as the end of the filament. Some intermediate filament proteins can form homopolymeric filaments, whereas others can form only heteropolymeric filaments. Vimentin, desmin, glial fibrillary acidic protein and NFL are able to form homodimers, which assemble into homopolymers. These proteins also can assemble into heteropolymers. In contrast, acidic and basic keratins, which form heterodimer with each other, form only obligate heteropolymeric keratin filaments. However, keratins do not form heteropolymers with other classes of intermediate filament proteins, whereas some intermediate filaments can. For example, vimentin and NF proteins can assemble into a heteropolymer. NFL self-associates to form a homopolymer, but NFH and NFM commonly co-assemble with the NFL backbone, so that most neurofilaments contain all three proteins. Whether an intermediate filament is a heteropolymer or a homopolymer probably is not dictated by simple hydrophobic interactions in the alpha helical segments in its constituent intermediate filament dimers. Rather, spacer sequences in the coil-coil sequences in the diverse N or C terminal domains, or both, most likely are responsible for determining whether particular intermediate filament proteins assemble into heteropolymer or homopolymer. Although intermediate filaments exhibit dynamic properties within the cell, they are clearly or more stable than microtubules or microfilaments. Remember, the stability of the intermediate filament presents spatial problems in mitotic cells, which must reorganize all three cytoskeletal networks during the cell cycle. Friends, you might be knowing how calcium and various kinases control the organization of the microfilament and microtubule in the cytoskeleton during the cell division. Similarly, under the control of CDC2 kinase, filaments of vimentin, desmin and lamens disassemble prior to or early in mitosis and reassemble after cell division. Without these sites, the lamin filaments remain intact and do not depolymerize early in mitosis as they normally do. In contrast, treatment of cells with drugs that inactivate protein phosphatases that causes rapid collapse of the network like lamin layer underlying the nuclear envelope. The opposing action of kinases and phosphatases thus provide a rapid mechanism of controlling the assembly state of intermediate filaments. Functions of intermediate filaments. Intermediate filaments are typically the most profoundly observed in epithelial cells. They comprise a structural integrity of this epithelia. Intermediate filaments are known to be formed out of proteins. They are the basic integral elements as skeleton of cytoskeleton. Depending upon the types, as discussed in the previous section about the detailed structure, the associated proteins have a particular and a typical role. As discussed, keratins of both type 1 and 2 serves for structural organization of epithelial cells. They are the most diversified classes of intermediate filaments. Hence, keratin isoforms are being expressed. This serves for a development of hard epithelial tissues that in turn forms exoskeleton as a protective structures like nails, hair and wool. Cytokeratins forms the basic structure that determines the functioning of cells present within internal body cavities. Type 3 intermediate filaments has individual role in certain associated cells like leukocytes, endothelial cells of blood vessels and blood clotting cells referred to as fibroblasts. The characteristic, if imbibed from these intermediate filaments, present as a part of cytoskeleton within these cells. Vimentin filaments also support cellular membranes. Their network aids the nucleus and other organelles in a well-defined place within the cell and remains in association with the microtubules. Desmin filaments of a muscle cells are responsible for stabilizing sarcomeres in contractile muscles to contract. The functioning of a glial cells and the structure base of astrocytes is only due to intermediate filaments. Remember, the function of peripherin is yet not known and studied in detail, despite of their presence in the cells comprising peripheral nervous system. A group of heteromeric neurofilaments as a type if an intermediate filament are responsible for the radial growth of an axon and thus is directly determining the axonal diameter. Moreover, diameter of an axon is directly proportional and related to the speed at which it conducts impulse. So, we can say that neurofilaments have an impulse conducting role as well. However, the influence of the number of neurofilaments on impulse conduction is highlighted by a mutation as found in quails named as quiver, which blocks the assembly of neurofilaments. As a consequence, the velocity of nerve conduction is severely reduced. Lamin proteins, as found exclusively in the nucleus, are responsible for the cytosolic location. The nuclear lamins form a fibrous network that supports the nuclear membrane organization. Familiarity with the various types of intermediate filament proteins not only is useful in comparing and contrasting their structural and functional properties, but also can help in the diagnosis and treatment of certain tumors. In a tumor, cells lose their normal appearance and thus their origin cannot be identified by their morphology. However, tumor cells retain many of the differentiated properties of the cells from which they are derived, including expression of particular intermediate filament proteins. For example, the most common malignant tumors of the breast and gastrointestinal tract contain keratins and lack vimentin. Thus, they are derived from epithelial cells which contain keratins but not vimentin, rather than from the underlying stromal mesenchymal cells which contain vimentin but not keratins. Because epithelial cancers and mesenchymal cancers are sensitive to different treatments, identifying the intermediate filament proteins in a tumor cell helps a physician to select the most effective treatment for destroying the tumor. Pathological conditions. In addition to scaffolding function, intermediate filament form complex signaling platforms and interact with various kinases, adapter and apoptotic proteins. Intermediate filaments are established cytoprotectants and intermediate filament variants are associated with more than 30 human diseases. Furthermore, intermediate filament containing inclusion bodies are characteristic features of several neurodegenerative, muscular and other disorders. The scaffolding function of intermediate filaments is best seen in intermediate filament deficient animals who exhibit disrupted cellular architecture, protein mistargeting, as well as alterations in organelle localization and function. In the case of lamin deficiencies, the impairment of nuclear composition has profound impact on many aspects of normal nuclear functions such as epigenetic changes, chromatin organization, or DNA transcription and repair. Intermediate filament variants are observed in various human diseases which reflect their tissue-specific destruction, whereas only few actin and tubulin variants have been described so far, likely due to their embryo lethality. Currently, more than 30 diseases are caused by and associated with intermediate filament mutations. Among them, keratin-related and lamin-related disorders are the best studied ones. For example, mutations in keratins cause a blistering skin disease termed as epidermolysis bullosa simplex, whereas mutations in lamins A and C result, among others, in different diseases including premature aging, cardiomyopathy, and lipodystrophy. The disease relevance of intermediate filaments is also highlighted by a variety of intermediate filament containing inclusion bodies, which represent the pathological hallmarks of several neurodegenerative, muscular, and other disorders. Friends, now it's a time to summarize our lecture on structure and function of intermediate filaments. As we have discussed the other two elements of cytoskeleton, intermediate filaments are also the important. The association of intermediate filaments with the nuclear and plasma membrane suggests that their principal function is structural. In epithelium, for instance, intermediate filaments provide mechanical support for the plasma membrane where it comes into contact with other cells or with the extracellular matrix. In epidermal cells, that is, outer layer of skin, and the axons of neurons, intermediate filaments are at least 10 times as abundant as microfilaments or microtubules, the other components of the cytoskeleton. Intermediate filaments are extremely stable. Intermediate filaments are found in the cytoplasm of different tissues and at the nuclear membrane as well. Unlike microfilaments and microtubules, however, intermediate filaments do not contribute to cell motility. The most ubiquitous group of intermediate filaments are the lamins found exclusive in nucleus. Epithelial cells express acidic and basic keratins. Unlike keratins, the type 3 proteins can form both homo and heteropolymeric intermediate filaments as vimentin, desmin, glial fibrillary acidic protein, or peripherin. The core of neuronal axons is filled with the neurofilaments composed of low, medium, or high neural filaments which differ greatly in molecular weights. All in all, intermediate filament subunits have a common domain structure, a central alpha helical core flanked by globular N and C terminal domains found typically organized in the cytosol as an extended system that stretches from the nuclear envelope to the plasma membrane. Thanks.